Hey, good afternoon to everybody that is listening to us for this different webinar from every week. Because today, instead of one person, we have three here, and we have a more long period to talk about coronavirus. So these pandemic situations that we live in out the world, and it's very different and it's very specific. But since many years, in vet side, we have coronavirus and work with coronavirus. So the idea today is talking about the experience that you have in the veterinary side with Dr. Diel, Dr. Maria Sayag, and then we have Dr. Sonia Villapol here. We're going to talk about the relationship about microbiota and the coronavirus in humans. So it's a different kind of a specialist that you have here. And all you guys that are listening, you can send questions anytime for us to discuss here. And I'd like to say thank you to all of you to be here and your attention. And I'm going to start with Dr. Sonia Villapol to introduce herself and talk her experience with coronavirus and how this starts in her life. Thank you, Sonia, for being here with us.
Yeah, thank you. Thank you, Elizabeth. So I am a neuroscientist, but my research has different focus on the inflammation and periphery. So as well, we are studying the brain-gut communication and how these mechanisms can, things that happen with the microbiota and the gut can affect to the brain and also to different diseases as stroke, ictus, concussions, and things like that. Also to Alzheimer's. So I know that the link with the vet is not very strong, but well, we have most of our experiments that are performing in mouse, in animal models, different disease. And of course, we need to consider always the role of the microbiome on the animals to translate that to the humans and to make the research that we're performing more translational. So everything that happens in large animals or small animals is very interesting for us. And yeah, we have also as well a clinical, several clinical research. One of them is related to the COVID and with COVID patients that they have inflammation in this kind of.
Okay. So let's. Hello.
Yeah. So basically, we look at the inflammation and also we have different treatments with antibiotics and with different treatments with prebiotics on these animals, and will be very interested to discuss with Dr. Diel and Dr. Assayag about that. I think that I'm going to learn more that explain, but will be interesting. Yeah, thank you.
Okay, thank you. And Dr. Diel, can you let us know your experience with coronavirus and what you have been research in this area?
Yeah, sure. Thanks, Elizabeth, and thanks everyone for listening. It's a pleasure to be here to talk a little bit about what we do with coronavirus. So I'm a veterinary virologist. I have been working with vaccine development and virus host interactions for the last 15 years. Since 2014, when porcine epidemic diarrhea virus emerged in the US, I have been also working with coronaviruses. One of the initial focus of my research with coronaviruses was actually development of vaccine candidates for PEDV, specifically for use in swine. So my lab actually works with vector vaccines, where we use large DNA viruses like pox viruses and herpes viruses as vectors to deliver vaccines in livestock, including pigs. So one of the first projects that I did in my lab, once I became faculty at South Dakota State University, was actually to develop a vector vaccine candidate for PED. So we expressed the spike protein of PEDV in one of our vector platforms and show that vaccine candidate induced protective immunity in weaned pigs. It was also able to induce passive immunity after vaccination of pregnant sows. We were able to show reduced mortality when we challenged piglets that were born from immunized sows. Recently, I have been, so my work with PED continued a little bit on, characterizing host responses and characterizing mechanisms of protective immunity, specifically targeting neutralizing epitopes, because neutralization is one of the important mechanisms of protection against PED and several other coronaviruses. So that was a continuation of our work with PED. And last year or this year, we started working with SARS coronavirus after the initial descriptions of potential spillover or spill back of the virus from humans into animal species. I also work with, in the diagnostic virology here at Cornell. I'm the director of the virology lab. So we started, developing diagnostic assays after the initial descriptions of potential infection in dogs and cats, so that we would be ready to diagnose the virus in case we would detect it in animals. Our lab was, we developed real-time PCR and validated those assays for testing of animal samples. We were involved in the first detection of SARS-CoV-2 in animals in the US, on that case in the Bronx Zoo where tigers and lions were infected. And since then, I've been collaborating with a group from NADC, the National Animal Disease Center in Ames, Iowa, which is a USDA lab, in assessing pathogenesis and potential susceptibility of livestock species to SARS coronavirus 2.
So very interesting. A lot of experience with coronavirus. And Dr. Assayag, can you tell us how is your experience with coronavirus?
Well, thank you for invitation. And we are working with coronavirus since 1995, 1997. Okay, that we started to see the many problems in the field with poultry breeders, and we always see problems with mortality, clinical signs, and the performance reduction. When we talk about the clinical signs in performance, we have a different situations in breeders and in broilers. Basically, when we think in performance, we think in egg production that affect the reproductive tract, the egg quality, because the tropism to the epithelium in the oviduct, and we see many different. We are seeing many different viruses in the world, basically with tropism to respiratory tract, reproductive tract, and urinary tract. Sometimes, and we can isolate, and we can reproduce the disease in the isolators, and we can analyze the tissue lesions with an immunohistochemical to see the viruses, and we see the clinical signs with a different situation, with different kind of viruses in different parts of the world. Like we've compared viruses from Asia, from US, from Brazil, with different situations in the field. We are seeing a lot of co-infections with different coronavirus in different tissues. In the same flock, we can see some some virus in tonsils, cecal tonsils and kidney. Another virus that prefer the respiratory tract. Sometimes we have in the same period, all the virus together. And another thing that I think that is important when we think in coronavirus is the protective type is more important than the protection against just one kind of viruses. Because the combinations with different vaccines increase a lot of the protection. We can see a broad protection for different kind of viruses. Sometimes we don't need one kind of vaccine to protect against another kind of virus that when we analyze the molecular sequence is very, very different, more than 6% in the S1, but we can protect with the combination with different coronavirus. And another thing is that we use live vaccines, attenuated live vaccines, different kind of attenuations with inactivated vaccines. All these combinations between different live vaccines and inactivated vaccines, we are working to get the best protection in the field, to reduce all the clinical sites that we are talking.
So nice. And I just remind people that is listening to us that you can send questions. We can discuss together. We can put you in the conversation as well. So we have here already Samuel Arivi send regards to Dr. Diel. And there is no questions, just regards. So when you talk about the coronavirus, I listen to you, I'm not very familiar with swine, but I know in poultry that like Dr. Assayag say, it's like a very recombinant virus. When I was like my first or second year at vet school, I worked in a project with this virus, and I remember that we do a lot of take samples from field and try to see what is the relationship between the different strains and their response. It was at the beginning, I will not say the year, but of course nobody knows so much things. And then they try to figure out together what is the protections that we have. So I would ask you guys, and anyone can answer, what is the difference that you see in the behavior of the coronavirus that comes from the swine or poultry, for example, with the COVID that we are having today? The main difference, I mean, in terms of behavior, can we think about it's the same difficulty to have a vaccine as we have in, at least in poultry, that we need to find a specific vaccine and they recombine every year? Or how can we expect this behavior? How is the mainly difference when you think about what, for example, Dr. Diel did or Dr. Assayag did to choose a vaccine for coronavirus in animals and to think about the development of vaccine in humans?
I think one of the main challenges is inducing protective immunity at mucosal surfaces. I think that's the main challenge with any coronavirus. I mean, with PED, there are several vaccines. I mean, PED was endemic and is endemic in Asia and has been circulating there for a while. They have vaccines. They have live attenuated vaccines. They have inactivated vaccines. The problem is, and in general, the live attenuated vaccines work better because you mimic the infection at the local site of actual virus infection and replication, which is the gut. The problem is most of these live attenuated vaccines are too attenuated, and that really doesn't induce an immunity that is strong enough to protect the animals when they actually see a wild type virus that is circulating in the field. And I think that's kind of the problem with most coronaviruses. With COVID, I think it's probably too early to draw a lot of parallels. But again, is a virus that will replicate in the respiratory tract, and probably the ability of any vaccine to induce mucosal immunity to prevent infection or reduce the infection in the respiratory tract will be important for the outcome and the efficacy of any vaccine that is developed.
Yes, I agree, Diego, because I think that the main point is when we in animals we think always in protection and partial protection, because the main objective is reduce the clinical signs. It's different than to give a total protection. And I think that basically the vets manage the disease in the companies, because it's different to I think that it's different to manage the disease to human beings, because we think in population, we do not think in individuals. It's an important issue to differentiate this point. Another thing
what you want to say is, like in vet side, like in animal side, we can think like we do a protection, then you're gonna lose some animals, and that's fine. But in humans, of course, if by ethical reason you're not take this decision, right?
And another thing is the local protection, like Diego said, because we work to protect to produce the local protection. Sometimes we need to use the vaccine, the live vaccine, I don't know, 2 or 3 times per year. And in the rearing period, we need to use sometimes 4 or 5 different vaccines in a short period and sometimes with different combinations with different vaccines to get the broad protection. The focus is always IgA. And when we think in the long-term protection, we can use the combination between live vaccines and inactivated vaccines, because we know that sometimes we can accept that inactivated vaccines, that we will have the challenge in the epithelium, that sometimes we will see some clinical signs, but we know that the reproductive and the urinary, we have the antibodies circulating, that the animal with that antibodies will survive, but we can accept sometimes some clinical signs. I think that's a huge difference when we compare animals and human beings.
Hey, Dr. Sonia, when you talk about, they said in animals, for example, in poultry, we have this effect in mucosa, and in humans, you have in respiratory mucosa. And when you talk about the relationship between microbiota and coronavirus, what is your hypothesis? Are you talking about microbiota, intestinal microbiota, or respiratory microbiota?
Yeah, no, it's very interesting. The previous commenting, I thinking about questions. Maybe we can leave with the questions later. But yeah, related with the microbiome is we are looking at change on the gut microbiome, because it has a direct effect on the immune system. And I was listening to Dr. Assayag before, talking about some kind of the mechanisms that related with inflammation with the immune system, and the way that this thing works. I was thinking that I hear that, for example, the bats, they have high levels, like they have some kind of the self protection system with the interferon gamma. I was that is some kind of the mechanism that they have development against all the kind of the coronavirus that they have in the past. So definitely is I feel that the immune system is very important, even animals or humans. This kind of the regulation and the gut is play a fundamental role on that. So even the recommendations to be protected is having a healthy gut microbiome, so you have a strong immune system, so you can prevent if you get infected by SARS-2, so unless you get less severity in the disease, and this is the major hypothesis. So of course everything is connected, and the levels of infection, the low viral, and everything can develop in different symptoms, and if we can manage the control the immune system is kind of the perfect prevention, to developing a worse severity of the disease. And I think that we can learn a lot about what happened on the animals to translate that to the humans, and should be more, related, and all these kind of the comments, they will be super. For example, another thing that we're talking about the combination of the vaccines. Right now in humans, we have 11 vaccines in the phase 3. I think that there are 6 with emergency approval, so that they pass. So, but of course, logistically it's complicated, but anyone is talking about the combination of the different vaccines, and they can, reform can be more protective, increase the percentage of protection, and nobody's talking about that. And it was something that was already experimented and was done in animals, so it will be very interesting. But well, logistically it's complicated because we have several limitations to generate and have vaccines for everyone, and even if we play with the combinations, will be more risky. But I am talking about the groups that are in high risk, like aged people or people with some kind of comorbidities, that they will be fantastic if we can think about a combination of the several vaccines that there are now in the last phase, and will be fantastic. And I have a question about the when you are talking about the co-infection. So it was proved in humans that people that get they get the flu in combination with the COVID increase the mortality in the 50%. So 50%, and that is huge. Like it's duplicated the high risk to die by COVID if you get the combination with the flu. And I don't know in animals if this co-infection with different viruses, they increase as well the mortality or the severity of the disease. That is another thing that also we can learn it from animals. I don't know, it's one question that I put on the table.
Yes,
but
of course, we have the same. I think that we have the co-infections, but when we have 2 situations in co-infections. The first one that I work with swine too, and swine and poultry, but in poultry we have a more problems with coronavirus co-infections with different strains that we can have a protection for one virus, and we do not have protection against another virus. And sometimes in poultry, the main focus is the protector type, because we have sometimes one protection small one with another one small one. When we combine, we have a broad protection. And another thing that we see a lot in the field is when we have a combination with like the coronavirus with I don't know, metapneumovirus, for example. That this combination we increase a lot the reproductive issues. It's interesting because all the viruses have a focus in the respiratory tract, but we combine both, we have a more reproductive problems than respiratory.
Interesting.
That's interesting.
Dr. Diego, do you have sorry, yes.
Okay.
Can go ahead. Dr. Diego, do you have some to add from your experience of co-infection?
I think it's always a problem with coronaviruses and any other viral infections, and in animals. I mean, there's a lot of other viruses and bacteria that will definitely accentuate or increase the severity of a coronavirus infection. I think one of the another example of a coronavirus that it's out there, is circulating in cattle, is a bovine respiratory coronavirus that we don't know. I mean, there's no data or no scientific evidence that that virus per se causes respiratory disease in cattle, but it is found very frequently associated with bovine respiratory disease complex, where you have other viruses and other bacteria. So I think that that's kind of a classic example where, we still don't know if it is the first virus that is there to, start the disease while, it replicates in the respiratory tract. Maybe it lyses the cells and then opens the door for other bacteria and other viruses that are there. Or it comes after, another virus like bovine viral diarrhea virus, and then increases severity. So it's pretty common, I think, and it happens very frequently with coronaviruses, where you see those cases where there is increased severity, and coronaviruses are just one in the mix of other pathogens that are there.
So I understand about what you comments here, that in animals we don't have 100% of efficacy. No, it's never we have 100% of efficacy in vaccines, but in the case of coronavirus, it's still like it's a lot of Dr. Assayag point out that the prototype is very important. The protection type is very important. So we understand that. Do you believe that for COVID we gonna have efficient vaccine? Like here we have Karin Franco from Brazil. She said that the Brazilian health authority said that the efficacy to have a vaccine for humans, it's at least 50% of efficacy. Do you think that will be possible, with your experience in animals, to have a vaccine with 50%, at least 50% of efficacy in humans?
I think that there's several factors that are at play here, and it really depends on which group are we testing this vaccine. And there's several clinical presentations with COVID too. So it's very complicated, and I think very early to assess that. Do I think it's possible to get a vaccine with 50% efficacy? Probably, and I hope so. But I don't know. I don't think I've seen enough data on the efficacy of the vaccines that are being on trials now to determine if the ones that are being tested are effective. I would hope so, because otherwise they probably wouldn't have gone into phase 3 clinical trials. So I would think that they do probably have at least 50% efficacy on those populations where they were tested. And for this virus, I think with the current situation, Any protection, it's good, right? We don't have a vaccine right now, so I think that we need something that will probably buy us more time to develop a better vaccine in case the initial ones are not as effective as we would hope for or wish for.
Okay, no, 2 weeks ago, I invited Dr. Jen Cook that works also with vaccine and poultry, but she was not available for today. So I had a webinar like 2 weeks ago with her. And I asked her if in her experience, there is any component of nutrition that could improve the efficacy of vaccine. And she said that does not see any relationship. So I'm gonna start with Sonia because you are testing like products for microbiota, or you are. Can you explain better how your connections with this microbiota and the clinical studies that you are doing right now with microbiota and coronavirus, and then later I'm gonna pass for the two guys here to put their opinion about how microbiota play with the coronavirus that they know in animals.
Yeah, we don't know about the vaccines as well. So the relation of the microbiome, the microbiota with, you know, with the efficiency of the vaccines, I don't think that there are a connection there. But yes, it's there with the immune system, of course. So the microbiome release several compounds when you have the appropriate bacteria in your gut. So we can be the healthy bacteria. We can call it like that, and so that produce interaction with some cells in your gut that produce compounds. Even some of them there are neuropeptides or some kind of the hormones or some kind of the nutrients that can help to the immune system. So that is the key, like the connection, the bacteria, the microbiome, and in the production of these compounds that can be good for the immune system. And with that you have the key of the disease that is basically is like inflammatory disease and since the beginning. So the theory starts because the COVID patients they have different symptoms, and the initial symptoms there are gastrointestinal problems. So they have vomits, diarrhea, pain, and abdominal pain, and all these things, and that happens just right after the infection, even days, 5 or 6 days before they develop respiratory symptoms or fever or other symptoms. So that was kind of a key, and that we find out that in the first from that the first report that come from Wuhan in February. So they report that some like 50%, 60% of the people they develop gastrointestinal symptoms before that they get, and that is very important because during this period, this time, the people can infect other people. So they are infectious, but they don't know that they are contagious. So they are, so it's very important attend to this, get the control and identify these gastrointestinal symptoms. And when we have inflammation or problems in the gut, so the microbiome, the microbiota, the bacteria that you have in the gut, sometimes they change, and some bacteria they are high levels and the other they are reduced, or the interaction between both change. And this is called the dysbiosis that also occurs in animals as well. It's the same. So that was the theory. We can block this kind of the alteration or initial alteration before development worse symptoms. We can reduce the severity of the disease because we're forcing the immune system. That was one the most optimist goal of our project. I will do something therapeutic, but this is so it's difficult because there are a lot of variables, of course. But the second one that is easy is identify specific bacteria that there are in different phases of the disease. So you can say that you have some species of bacteria, so you have a high prognostic to development more or less severity of the disease. So, and that basically is the thing that we are doing in my hospital, and as well we have a collaboration with another hospital in Argentina. So they have patients that we analyze the microbiome in the beginning when they go to the hospital, when they're hospitalized, when they are leaving the hospital, and then during this period we look at the microbiome of the bacteria they change, and we correlate all this data as clinical data. We correlate that with the age, with the gender, with the severity of the disease, with the development of other pathologies, and we try to identify the key bacteria that we can later modify or modulate the some way to find something more therapeutic. And I feel that this is important because the gut microbiome is not explored at all in most of the disease, and the COVID cannot be a exception. So far, there are a few publications that they show some bacteria change on these patients, specific to these patients, but it's very, because the disease doesn't have, it's not that we are studying that for a few months. So I hope that we can get better studies later.
Yeah, you can say something about that the symptoms changes because of different microbiota by now, or it's just a hypothesis?
No, not for different. The virus. That the entrance is to the angiotensin, the enzyme convergence for the angiotensin 2. That is the ACE2 receptors, and that the receptors that are expressed in most of the cells, some of the organs, the concentration is higher, and the intestines, they have a high concentration of the receptors. So not only that the virus goes through the respiratory tracts, but the circulation is on the blood, and feeds into the circulation go through all the organs, and the intestines is another target. So there, the virus can infect the intestinal cells, and they can induce inflammation there, and direct change of the microbiota there, and as well the systemic inflammation that the virus causes. Of course, if there are the cytokines stored in this like a high inflammation, systemic inflammation, of course that is a general change that can affect the whole body, even the brain. Now is the last thing that we are find out that that as a neuroscientist, I am very interested in everything that happens in the brain, in the gut-brain connection. We know that, but now we are having patients in the hospital that they are recovered patients, COVID patients. So, but they come to the hospital with neurological problems, or even stroke, or like anxiety, depression, and things that they were or things related with neurological problems that were not expected, and even they have this fatigue and the loss of memory, and this kind of the problems that affect to the brain in some way. We don't know exactly why this thing happens, but
Mechanism.
because in these ACE2 receptors in the brain, I was studying that since I started to study ACE2 receptors in the renin-angiotensin system with hypertension and 10 years ago, and I know these mechanisms, and I know that the brain, the neurons, they have a few receptors, a few of the receptors for ACE2. So it's not that a strong possibility that the virus directly go through the brain and infect the neurons there, but probably the secondary effect that is systemic inflammation, or can across the blood barrier if there are some kind of the problem there, so the virus can do it, and that is a little bit worrying. The long-term problems, neurological problems that the virus can have, and specifically neurodegenerative disease, Alzheimer's, Parkinson, and can be possible. It's so early to say anything, but for example, was one case identified the first patient that developing Parkinson's symptoms after get recovery, and was perfect fine. So it's only one case, it's nothing, but we are in the month 9 since that this pandemic started. So we'll see what happened, what we have in 3 or 4 years. So that is a little bit worrying. And we, it's important that the vaccination work as soon as possible and get the people protected as better that we can do it. So we can not just prevent the deaths, It's just prevent the
Yes.
long-term effects and all these kind of the problems that we don't know now, who can be.
What's gonna happen? Yes,
Mhm.
I read an article this this week in Nature that they find when they put microbiota that they have immunity against another bacteria that go to the blood that was not the one that was in microbiota. So they show finally the hypothesis that when you have a microbiota, you really make a modulations for answer another vaccinations that another antigens that is not the one that start with the microbiota. So
Yeah,
this is, I think, it's very interesting point, and this is why I was very interested in your studies about that. We have questions here. People start to ask a question. So they ask about the herd immunity. Samuel asked it. He took a course of with Diego, I think, and you talk about herd immunity. And people are talking about herd immunity now. For example, there is a study from UK that also was published this week, I think, that they said that when you have not lockdown but let the young people catch the virus, they increase the herd immunity. And according to their results, the best idea is to lock down just the sensitive people, like the people with low immunity or old people, and to let all the other populations, especially the youngest one, to catch the virus. So how do you guys think about that? Maybe start with Diego, that Samuel asked to talk about herd immunity.
I think herd immunity is critical for any infection, right? At least to reduce the circulation of the virus. The problem with SARS-CoV-2 is that we know so little about the virus that I think would be really risky to use that strategy to contain the pandemic because we don't know much about immunity about this virus or against this virus and what is actually really required to have herd immunity. I mean, I think now there's a few examples of places where, like New York City, for example, where the virus had a really wide circulation because of the population density in New York City. And I can't remember now if it's 60 or 70% of the people that actually did antibody testing, they tested positive. So it's pretty wide circulation of the virus in the population. And that the fact that the virus is not circulating anymore suggests that herd immunity helps in reducing that. Um, the issue is how do you isolate only elderly people or people at high risk groups, people that have comorbidities and all of that? It's a little more challenging. It's not only the age, but you do have people that have underlying conditions, and a lot of them don't even know that they have an underlying condition that would increase the risk of more of death. So it's a challenge to balance that, and I think it would be really difficult to adopt such a strategy in humans. I think it's a lot easier in animals because you always have animals are raised in age groups, right? So it's easier to adopt such a strategy in animals than it is in a very heterologous human population. So I think that's the challenge with this virus. I don't know how much benefit it would actually really uh, bring. And I think there's a few examples that, England, I think, was one of the latest countries in Europe that decided to close, shut down. Their idea was exactly the idea of herd immunity in the beginning. And then they started, seeing a lot of mortality, a lot of death. And that's when they went back and revisited their strategy and decided to do the lockdown because it was working in other neighboring countries.
Yeah. I don't know. Can I go now? Yeah. So, Elizabeth, can you hear me?
Yes,
Yeah.
yes.
Yeah.
Oh, sorry. I
Yes,
didn't.
yes.
Yeah, no,
Yeah.
I feel that this is kind of a risky statement, and even yesterday was published in Lancet a letter signed by 80 scientists, and they make this claim that the herd immunity is a dangerous fallacy and supported by scientific evidence, and it's kind of true because as uh, Dr. Diel mentioned, so some countries they decided to do that and they failed. So, for example, Sweden, they have like, I think that they is the second country in the world they have more mortality by COVID, and they didn't take any measures. So they no mask, no social distance. So they tried to this approach, and I think that is going to be the ruin of the country in the long term. So that the mortality was anyway, it's the same that we were talking before. The reinfection is a fact, can happen only. I think that were detected as less, only 20 people. It's not that important, but they were some people that they get infected with one strain of the SARS-2. They got infected later, so the reinfection can happen. Even it's very anecdotic, and the cases there are a few of them, but this is one point. So you get infected, but you can be reinfected. And then another thing that is critical, how long will last the immunity? If you get infected, you can generate antibodies. The last study was say that you can identify antibodies and immunity under 4-6 months, but then after that you are in risk again. You are not protected, and only the vaccination can do that. So that is in other things that the other number that they say is that, and I agree that there are the 30% of the population are vulnerable. So there are all people or people with comorbidities like diabetes, obesity, and all this cardiovascular disease, hypertension. So it's a percentage of the population that will be exposed, and with no effective treatments, they are at risk to die. So that is not. It's crazy, analyze and propose that. So some of the countries that they don't take all this kind of the preventive risks to protect themselves against the infection, they are in high risk to, that this percentage of the population, they're going to have long-term consequences really, really bad, because when they are recovered, the people they get out of the UC and they get recovery, like 90%, they have problems later, and we don't know for how long will be. So there are a lot of factors that, in my opinion, and apparently this statement from the scientists also, and almost everyone is saying that this is a crazy idea and doesn't have any evidence that this thing is going to work right now. So we need to wait for the first of all protect ourselves as much as we can, and take all the protection measurements like that we're heading in the past months, and then the vaccination to the population is very important. They had to trust in the vaccines, because if the people start to be scary about the secondary effects of the vaccines because they don't believe in science in this way, I suppose that the vaccines that they go through all these phases, they're going to be self-eating, efficacy. So they have these two things that we have to consider. But I feel that it's very important that the people don't have this thing in the mind that it's good get infected everyone, because it will be a catastrophe. Like, I don't think that any country can can survive with this idea, and the proofs that are there in these few months. So there are some countries they definitely fail. And as Dr. Diel said before, so some of them they change the policy, the politics again. So as UK, so yeah.
I think, New York City is kind of an example of that, where there was lockdown, there was shutdown of everything. But because of the population density, still a lot of people ended up dying because the virus was circulating like wildfire. I mean, you have 10,000 people per square kilometer in Manhattan. That's probably the highest density in the world. And it's really difficult to contain a virus that spreads this quickly when you have so many people living, in high apartment complexes and high apartment buildings. And even with the lockdown, a lot of people ended up dying in New York City during the peak of the outbreak. And I'm Brazilian, and I think Brazil is one of the countries that is failing and failed in not really following the lockdown rules. And we have seen, and we are seeing, Brazil is the second country in the world in number of cases and in the number of dead people. And that's because people did not follow the lockdown rules, and there was a lot of, it was politicized more than it should have been, rather than listening to, people that understand infectious disease and that have knowledge about that. I think there's a lot of politics going on that really complicated the whole containment of the situation in Brazil.
I think, Betch, I think that in poultry, we have many data about the herd immunity. And it's important that always we have two different situations. The first one in breeders, when we have a problem in a farms, when we have some farms near many farms in the same area, when the virus starts in one farm, after usually die 5% to 10% of the flock, that's a lot. When moved to the second farm, reduce the mortality to 3 to 5. When moved to the third farm, reduced to 1 or 2%. To the fourth farm, we do not see any kind of mortality, and we can see only clinical signs. And after 5, 7 farms, we can see that the disease will losing not power, but we will lose we reduce the clinical signs. And it's always have exactly the same situation, the same way. In another, when we're thinking about the herd immunity, it's very easy to see in the processing plants too, because when we mention broilers, it's much easier to see two factors. The first one is feed conversion, and in the field, and the second one is the condemnation in the processing plant, because we can see we slaughter all the poultry, and in the processing plant we can do the crops in 100% of the population. It's different than humans that we cannot do. We do not test 100%, but in broilers we test 100%. Every 40 days, we can see exactly what is going on in the processing plant. And we have basically two kinds of waves. We have one short wave that we have a very high peak, that the period usually is 3 months, between 2 and 3 months. Why 2 or 3 months? Depends on the cycle of the broiler, because the cycle is 40 days. If we have a griller that the cycle is 30 days, the peak is very short. When we have a heavy bird that we slaughter every 45 days, we have a 3 month, 2 cycles. And another thing is when we have in some regions that we have some low density and low pressure, the peak is not too high, but the period is around 4 to 6 months. And it's very, very easy to see, and I published some paper about that, and you can see in the field, because all the companies have exactly the same situation. Last week, we went to west of Paraná 2 weeks ago, and we saw exactly the same with coronavirus in processing plant. And always we have these two situations. In breeders, that a long cycle, that's 60 weeks, we can see the virus moving in the farm. And in broilers, we can see the results in the processing plant. And it's interesting because we do not have, sometimes we do not have any protection against with vaccine, but we see the same condition in the field. And and you can see basically the same in all parts of the world. You can see the same kind of the kidney problem that you see in China. They have the same curve with 3 weeks, 3 months. In Brazil, respiratory or nephritis problem with different viruses, we see the same curve. And sometimes I think that the people are thinking more in humans, that in humans too much different than the animals. I think that have a more relation in herd immunity than we can imagine.
Can I just make a comment about that? I think there is an important difference that we need to keep in mind, is that IBV is probably circulating in flocks that have some immunity, right? So that immunity will actually put pressure on the virus that is circulating in one flock, and then it goes to the next flock, and then that virus is probably a little bit more attenuated because it has gone through a flock that had pre-existing immunity that induced change on that virus due to immune pressure. Now we're talking about COVID, where basically the entire world is naive, has never seen that viral strain. So there's a difference there. And I don't think we have seen a lot of differences in the mortality rate as the virus spread throughout the world. If the virus infects the people that are at high risk, the mortality will be high. So, and that's also seen in terms of genetics of this virus. Is basically the same strain that is circulating, the same strain that was detected in China is circulating in the entire world, with the exception of a few nucleotide changes across a 30 kilobase genome. So it's a stable virus. There's not a lot of change. There's not a lot of change in virulence and pathogenicity because we are all naive to it. And that plays a big role in terms of deciding on how to do what needs to be done in terms of allowing the virus to just ramp up free across an entire naive population when we're comparing to an animal population. I think that it does play a significant role there, and I think that there are significant differences in those two scenarios.
There is some questions here related. I saw you guys talking about sensitive people, and there is questions about this here. For example, some questions about studies say that obesity or overweight represent the same risk factor as age. Another question here that there is any relationship between blood type of blood and less susceptibility to SARS-CoV-2. So can you comment on it, like what makes these people sensitive? And first, it was any relationship between the blood type and the SARS-CoV? And yes, they ask also if in animals we have the same, like the blood type makes any difference.
I can respond to the obesity you want because it's more related with microbiome.
Okay.
So the obesity is a comorbidity of the COVID. Everyone's know that you are obese. You have high risk to a worse prognostic and development of the severity of the disease is worse, and that is related again with the microbiome by one way. That is something that is we are considering that because the people that is obese, they have microbiome dysbiosis. The dysbiosis, there are the alterations, the change on the bacteria in the gut that usually they decrease the diversity. So we can say that you have more bacteria that they produce bad metabolites, bad compounds, and you have a few bacteria that there can be the healthy one. So there are dysbiosis, so there are these perturbations in the gut of people that is obese. So that means that the immune system is reduced and is not strong. And as well, another thing that is another hypothesis in parallel, because again it's related with the immune system, is that the adipocytes, that there are these cells in the fat that that fat, so they are very inflammatory cells. So they produce a massive amount of the cytokines. So that increase the cytokine storm and increase the inflammation on these people. Even if you doesn't have to be a morbid obesity, it's like just a little bit obese. So it's important the high levels of the inflammation on these people for these two mechanisms, the bacterial dysbiosis and also the adipocytes that they produce more cytokines.
I just have another question that maybe be together with microbiota. There is another question here that some specialists, they say that we need to take vitamin D, and the vitamin D has a strong immunity against COVID. What do you believe in it? And it's a direct effect to the vitamin in the host or the vitamin microbiota in host?
Yeah, this it's not I believe, it's the only few things that they were, there are data and there are publications that they support this idea. So the vitamin D can reform the immune system and the people that taken the vitamin D, that they respond better. So that is again is related with the immune system. So you need to get this compound, this vitamin is crucial for the immune system and to be strong with that. But yeah, the vitamin D is true. There are a lot of things that people say, the prebiotics, but there are no data that report that. But the vitamin D, yes, that the CDC they accepted and then was considered one of the compounds that we need to have, high levels in the diet to be protected.
So, but do you think it's because of the cateocytin that is produced, like it's an innate immune response that helps, or it's like the microbiota that makes some metabolite? How do you think that could be the mechanism on it?
Yeah, all then it's multisystemic. It doesn't affect only one system. So yeah, the vitamin can affect the different systems, but directly in general is a systemic inflammation. That reinforces the immune system. Different pathways can target different pathways, not only the gut, but the gut as well. Mm-hmm.
So do you guys, Diego and Mario Sergio, do you think that the vitamins or something like it they find in humans could help in this disease and in animals? We need to think about something like that. You have some experience with it?
Mario, go ahead. I don't have a lot of experience.
I do not have experience with that, because what's going on is that our feed, we formulate the feed for with the not the perfect, but we have a different situation in animals, in production animals, because when we formulate the feed, we have all the minimal levels that is necessary to achieve really good production standards. And when we have some deficiencies, we can see the results in the field very easy. Like if we have any kind of vitamins the deficiency. We can see problem in the processing plant in skin, in foot pet lesions. We can see a lot of things, and in breeders we can see the feather problems in swine, skin problems. It's very easy to see the results. I'm sure that we, if we do not have the minimal level for all the vitamins and microminerals, we will have problems in the population, like we can see in the processing plant.
Yes, but
Yeah,
for example, studies. Yes. No, I
yeah,
just
no,
gotta say that this.
The thing that I hear about the the nutrition, animal nutrition, and they is now in the market. They play an important role, the probiotics and the prebiotics, and because they the microbial metabolism, and especially the food for animals that they contains the high fiber diets that increase the immunostimulatory short chain fatty acids, that there are the compounds that can protect and make strong the immune system, and that is mediated through they make strong the lactobacillus and the bifidobacteria and the gut of these animals, so they are more healthy and they have less disease and less problems. And I know that there are the diets and the animal nutrition, they are incorporating all these compounds that they are related with prebiotics, not probiotics, but both. I feel that is something that the nutrition they should consider as well, because the same that happened with humans, and it's important for all the disease, get the microbiome and the nutrition under control, and know very well what kind of the food can cause the dysbiosis, and that can alter the immune system and make the animals be sick. So I think that it's important all these considerations and the animal nutrition, but and also on the market will be like explosion. I think that in the future years that will be generate a lot of money also for the companies that they make this kind of the foods for dogs and cats and also farm animals. So I feel that should be important.
Yeah,
I totally agree.
About vitamins, I will not completely disagree, but I just have these studies from Dr. Claesing that he said that however we have the level of vitamins and ingredients, not just vitamins, minerals, and we have it for the levels for productions, but not for immunity. Maybe sometimes you don't see deficiency in productions, but maybe when you have like more inflammation, you need more antioxidant. And then this is what he said, and he shows some data in that we sometimes need some improvement because of the antioxidant effect demanded by the inflammatory effect. And there is also another question here in the same area where they talk about if what is the opinion to use plant extract that is antioxidant for this coronavirus inflammation process. If some plant extracts that are antioxidant could bring some advantage to use for COVID or for coronavirus in animals in general. Silence.
I can't talk for humans, but
I don't know, because usually we when we have a clinical issue, we see a clinical problem, it's difficult to think about. It's different to prevent, but when we have the clinical problem, it's difficult to think about use this kind of things. Not my opinion.
But
really
that is there is to another question, another opposite way, not about a specific plant extracts or prebiotic. However, I'll pass the words for Sonia after, just to finalize. There is your, like, in the animal side, do you have any information about if the coronavirus in swine or in poultry, they are more sinus because the animal has different kind of nutrition? Do you have any information about that? That the sign or mortality it's higher because the nutrition? There is any information that or not?
Can I say? When we think about the microbiome and we think about the environment, I think that's important because we basically we have two kind of challenges. We have when we have like a third enteric coronavirus, they cause a very strong disease with this epithelium destruction. It's a real it's a clinical disease, very strong inflammation and very that destroy all the intestine. Okay, that's one case. Another case is like we talk about the Runting-Stunting syndrome. When we think in syndrome, that we have the enteric coronavirus in broilers, that we can see the virus in the intestine. Of course, that the microbiome is very important, and I think that we can differentiate. See different kind of the challenges. When we have this kind of a syndrome, that we have a multifactorial syndrome, that all these issues, that all these points is really important, and when we protect all the sites, we will have more success, and we can see that in the feed conversion and the number of flocks that we have the disease. The microbiome is really important. To have a better environment to protect against this kind of syndrome, or reduce, really reduce, and a good low pressure really reduce the clinical signs.
Yeah, I have a question maybe for you three. They can respond because I don't know exactly. So that related with that. So we have experiments with animals in my lab, and we use rodents usually. We have also pigs for a big study, but for the microbiome studies, we have mice, and we have several problems when the treatments that we have for the microbiome. So we do prebiotics, and we do also probiotics. We grow some specific bacteria, usually the lactobacillus or the species, or bifidobacillus, it depends. So and then we administrate these probiotics, and we have another. The last one challenge that is as treatment is the fecal matter transplants. So we transfer the poop from some animals, and we restore completely the microbiome from other animals that they have Alzheimer or another disease, another animal model. So my question is that probably you have more experience with that. What about the antibiotics? Because we have different groups, and the antibiotics for the animals. So usually is to kill all the bacteria, that we have a cocktail antibiotics, and we shoot the antibiotics, and then in theory we eliminate the bacteria, so the new bacteria can colonize the gut and have better function, or the colonization can be more efficient. But we don't know exactly this kind of because we're killing everything. We're killing the good and the bad bacteria with antibiotics. So what is your experience using antibiotics in animals? Just to relate with inflammation and related with the health in general, the negative results, of course, not when you are using antibiotic to kill some bad bacteria, but what kind of negative approach or you can think about that? Is really bad, or use a cocktail antibiotic systemically always and doesn't, that you are killing the good bacteria that can have beneficial effects, but as well, you don't restore this bacteria with new bacteria. That's me. This is my question. What are you doing with that?
I have very little experience with this, but I think, the few publications and presentations that I've seen, the use of antibiotics in feed, at least in swine, they do have a beneficial effect, even though they are reducing the load of the good bacteria. They keep the bad bacteria on check, and I think the balance between the bad bacteria and the good bacteria is even out at the end, and it ends up helping productivity, right? Because they're used basically to enhance productivity, and I think that's my opinion. But I also share concerns about wide use of antibiotics, and we have been using them for several years. So I think I recognize that there are problems, especially in introducing resistance to those antibiotics in animal feed.
That is in combination with the administration of probiotics after antibiotics, or you don't do this practice in vets?
No, in fact, what happens, I'm just going because, in fact, Jeffo is one of the companies that work to reduce the use of antibiotic in animals. There's many companies, because the idea is that in the past, and I agree with Dr. Diel, we use these growth promoters, antibiotics in low lethal in the diet, and they really improve the performance. Maybe Assayag will talk a little bit later, but the situation is that to modulate the microbiota, we don't kill all the bacteria, of course. It's just to modulate it. And there is some few studies that shows that these antibiotics, that is macrolides, they are also anti-inflammatory. There are some studies that show in vitro that because you use it, you have less inflammation. Because you have less inflammation, you have better absorptions, and all this gonna help to the growth and performance. But in these days, we believe that because of all the resistance that could happen and the problems that use antibiotics in feed mill and everywhere, the idea is to control it. And because you have another options today, like have mentioned plant extract, organic acid that you mentioned, prebiotics, probiotics, all these, it's today giving the same results that you have with antibiotics. So in the animal side, what we look is like to try to avoid use these antibiotics, because we have another kind of products like you mentioned that give the same results and associate with biosecurity and now the welfare with the animals. So this is our idea in the vet science and animal science today about that. And this is it's why. But if you ask, yes, we use probiotics and prebiotics associated to antibiotic as well. But the idea is to avoid just using disease and not as a increasing performance.
When we think about antibiotics, I think that we need to think in two things. We need to think in systemic antibiotics and enteric antibiotics. The first step. The systemic will have a positive and negative. And enteric, we focus basically in positives. And when why we see this gram positive with only enteric action, the better feed conversion and carcass yield. Basically, like Dr. Santin said, reduce modulate the population, the bacterial population, reduce the inflammatory condition in the intestine, and reduce the feed conversion. because increase the efficiency to use the feed. And of course, it's not the past, not is year after year, all the companies and the countries are moving to other products, are focusing therapeuticals more than growth promoters. I think that's the main point, and moving to acids, like Dr. Santin said, prebiotic, plant extracts. And the focus is to get a better enteric conditions to reduce the feed conversion and increase the health. But sometimes when we increase the immune response, we increase the feed conversion, and we want to reduce the feed conversion. If the animals stay in a very good environment, when the animals spend too much in the immune response, in the immunity, we will increase the feed conversion. But if the animals stay in a bad environment, sometimes this immunity that some products can improve, the feed conversion will reduce too, but it will depend on the environment and the microbiota, of course, because it's important, because sometimes we think that different products, we can see synergy, but we sometimes we do not see the synergy that we expect. And sometimes we do not expect synergy, but we can see in the field the result when we combine prebiotic, probiotic, and acid or plant extract. It's very interesting. It's more difficult to preview the results according to the microbiota.
So we are almost going to the end of our meeting. It's a lot of questions, a lot of things to do here, but I like these questions here about what will be the directions that the research should be facing to come up with an effective vaccine for COVID, I mean. So you guys had to work in animals with vaccine, and Sonia, about your experience, what you have observed in your clinic studies with microbiota. What you guys think that should be the research should facing, like there is any some research that show that kids should be vaccinated before, and others that say that the adults should be vaccinated before. So you can address all these situations for people that are listening to us.
Well, I think that the kids, fortunately, the kids, they have minor mild COVID or even symptomatic, so they get infected and they have a lot viral, like enormous, like a lot. They have a lot of virus, but for some reason, well, there are some reasons. There's the innate immunity that is higher in kids, so they are not in risk. So the vulnerable people, the old people and people with vulnerabilities, they are the ones they should be vaccinated first. And also the companies, I think that they should try, these vaccines that are in the phase 3. So they should try more in older people. And so far, I think that only Moderna with the RNA vaccine, this company, they put in all people, and it's great. And then they respond well. It was as only 40 people, but the results, they were the same that adult people, young people. So they produce antibodies, neutralizing antibodies, and they have immunity in response to the vaccination. So that is great news. So I think that, of course, the vulnerable people has to be the first one. But also the protocols or the companies that they produce all these vaccines, they need to be transparent, and the protocols, they have to have the endpoint. and where the symptoms and everything, that also they have to include the old people, vulnerable people. That's the matter. Everyone, we can reduce the transmission if everyone is vaccinated, but of course, the old people, they are the people that need to get protected. If we need to have a vaccine, it has to be for the vulnerable people first, and then we can protect the rest. But yeah, and of course, the kids, they need to be vaccinated as well. But I think that is the risk on the kids is the transmission, because they can transmit the virus, right? So, but they don't have, they doesn't affect that much to them. So I think so. That's my opinion.
I agree with that, but I think the key obviously we need to protect first who is at risk, right, or a higher risk of death. But the ideal vaccine for this will have to really prevent transmission. So I think that's one of the key factors of any vaccine to be effective against this virus, is really reducing virus shedding and transmission, because I think that's one of the key features of the virus that allowed it to spread the way it did across the global population. So the vaccines will really have to focus on reducing virus shedding even in asymptomatic people. I think, obviously the issue whenever the first vaccine is out, the main issue is how do you produce enough vaccines to immunize the entire world, right? So I think that prioritizing first doses to the people that are at risk of mortality, I think that's obviously something that needs to be done. But in terms of vaccine design and research, it would have to be a vaccine. The ideal vaccine is a vaccine that will actually reduce transmission of this virus.
I agree. I think that the point is how we will have a herd immunity, and I always thinking that when we have one coronavirus and we vaccinate all the population, we after 5 years, 4 years, we will have another coronavirus. That's the point that we are seeing and I think that when we think in research, we need to think not just to protect, we need to have a better vaccine that can be easy to combine to produce a new one that because I think that if we learn with animals, we need to be fast to register the next vaccine to protect the population.
The corona.
Yeah, and
Yes.
I think that, regarding to that, and that Dr. Diel say as well, it's super, the transmission is the key, but since at the beginning, they were talking about the social vaccination. And the social vaccination is just protect ourselves the best that we can, until that that the vaccines that we have now in phase 3, they are produced and they are, administered to everyone, but that is going to take years. Probably, the people that is not in risk, maybe a couple years or 1 year more, who knows. But, that the social distancing, the use the mask, that is something that we need to live with that for a couple of years or less, I think so. And also it's very important to know the risk and the risk of the transmission, as Dr. Dial say, that the people say that the distance is important, but we know now the revolution of that the scientists, they were saying that for a long time, and it's difficult to believe, but the virus can be transmitted through the aerosols, and that is the thing that we need to avoid, the indoors and the places where there are a lot of people for a long time, and we need to do the ventilation has to be a priority, or use filters or other mechanisms. So that we need to do this this social vaccination so far until get something, to get something better and be everyone protected. Yeah.
Yes, I agree.
So to finalize, I would like, of course, to thank you everybody and everybody that is still listening to us here. But I'm gonna leave this time for each one of you give the final message, what you want really to take to let these people to have as a final message about coronavirus, COVID, whatever that we discuss here today. Let's start with Diego.
I think one important message I think that this whole pandemic taught all of us probably is that research is really important. You do have to invest in research to be prepared to deal with new pathogens, not only viruses, but bacteria as well. And I think that investing in broad use platforms, as mentioned by Dr. Assayag earlier, that a platform, a vaccine platform, for example, that can easily be changed and adapted to any new emerging pathogen that will come up eventually. It's important, and that can only be done with research and with investment from the government and from the public too, because it's public funding that will fund scientists like us to keep doing what we can do to help on fighting those new pathogens.
I think that, like Diego said, I think that we need to use this research, all the research that we are, and the situation that we are living right now to learn to protect better our animals. I know that all the information that will be possible to use in the next years will reduce the time to produce the new vaccines to be much faster to have solutions to this kind of situation. In my point of view, I think this in the final, after 2 or 3 years, I expect to have this to faster capacity to produce good vaccines to protect the people, not only to respiratory or coronavirus or influenza virus, all the vaccines. And another thing is to have this information to produce better vaccines to protect the animals.
So I will send the message to the humans. So I agree with everything, and I feel that the message should be that that we had to trash the investment that the companies, the governments, and the scientists they did on the vaccines resharing, all the vaccines that there are now in phase 3, that we need to have be patient that they will be safe and they will be efficacious, and we need to believe of that because they are doing the things with the right protocols, and the protocols they need to be transparent. So we have to trust the science. And regarding to the herd immunity, that I just refer this paper that was published yesterday in the Lancet, that as I said before, that the herd immunity is a dangerous fallacy and supported by scientific evidence, and we have to avoid that and have so far this social vaccination and reduce the transmission as better as we can, and hope that that the vaccines can arrive soon to everyone. And that's it. Thank you.
Okay, thank you so much for all of you for this time and for the good discussions. I wish you a very good evening or night or afternoon, I don't know. And thank you so much for you who are listen to us. And we have some questions here. Maybe we just share with you three, and you can answer if you want. Okay. Thank you so much. Have a good day. Bye
Thank
bye.
you. Nice to meeting you.
Thank you.
Nice to meet you. Bye bye.